Welcome back to another episode of Professionals Podcasting. This is another in the series of Flying Solo with Attorney Bernie, where Attorney Bernie discusses the brain with Gary Howard and Matthew Kaiser.
What do we know about the brain? How does it work at a cellular level, molecular level? What we don't know about the brain, and how are people modeling the brain right now? Where we are in modeling the brain, how this correlates to computer models, and lastly, we're going to be talking about organoids. Now, when I saw that on the list of topics, I was intrigued, and I'm worried that we're not going to have enough time to get there. So why don't we just start there first?
Of course. So what's now being done in many laboratories around the world is that scientists are being able to take stem cells or cells from somebody's body and tune them up you treat them with different chemicals and molecules and growth factors, and they turn into a generalized, what's called a pluripotent stem cell, and that can be changed into any particular type of tissue. And so what they're doing now is they're taking those type of stem cells and they're able to grow them in a three-dimensional pattern structure, and the cells just divide and then they form connections with each other, and they're essentially forming a smaller organ version of what the cell type is. For example, you can make muscle cells into muscle organoids, and heart cells into heart organoids. And in this case, we can make neural cells, neurons, and other brain cells into small mini brains.
And what's the practical application of that, Matthew?
You can, by that way, you can then study how the cells interact with each other, and we'll actually be able to understand how the cells communicate with each other on a larger structural basis. So, for example, these organoids, can be a sort of a small bundle of cells of several thousand cells in number, and they essentially sort of can communicate with each other. And so that way we'll be able to understand a little bit more about how, for example, as we're going to talk about it later on, how brain cells operate. One very interesting piece of research has come out recently within the last year, is that people have been able to grow brain organoids from both chimpanzee stem cells as well as human stem cells. And it's interesting that when you grow these organoids up, the human stem cells always grow into an organoid that's about twice as big as the chimpanzee one. So, and we know that from our obvious studies that our brains are 2 to 3 times larger than those of chimpanzees, and so there is something in the difference between human cells and the chimpanzee cells, which is dictating that the cells grow more often or more repeatedly. And some people have now found that there's a particular gene which, when it's switched on in humans, it's the human version, it actually causes the neurons, the precursor brain cells, to divide 2 more times. And so your starting material is 3 to 4 times more cells than you would have than otherwise. So that's how you end up with a larger brain.
Well, it seems as if we're getting ahead of ourselves here. So, Gary, why don't you just cover what do we know about the brain right now? How does it work, and what don't we know? We'll just cover that, and then we can talk about the modeling and the computer models.
Glad to do that. The brain and how the brain works is really, I think, one of the last major problems in biology. Gene regulation, how genes turn on and turn off, is pretty well understood these days, and a lot of developments understood, but how the brain functions is one of the last great problems. So I do know a lot about it. I mean, clearly it's a physical entity, and it depends on a lot of chemicals as well. And we know that because injuries to the brain destroy the ability of people to undertake certain functions. Like we know that a stroke or physical damage on the left side of the brain often interferes with people's ability to speak. We know that drugs taken therapeutically can help with various kinds of mental illnesses and so forth. So we know that there's a real interplay between the physical side and the chemical side, influencing how we think and behave and so forth. We know a lot about that from the molecular end, really. We know that a particular kind of cell called neurons are one of the major players in the neural system. There's a lot of them. There's nearly 100 billion of them in our normal brain, and they are connected with each other and with muscles. And we understand how the electrical impulses that are related to thoughts and actions are transmitted along the nerves and transmitted from nerve to nerve and from nerves to muscle. We understand the little chemical interchanges called synapses or neuromuscular junctions. We pretty much got all that down. That's fairly well understood.
So, Gary, where do we go from here? What don't we know?
What we don't know, though, is if you remember in DNA, there's a series of 4 chemicals with the names A, T, G, and C, and they're in a long sequence, and they encode the proteins that make us up. Right now, all of these billions of neurons in our brain make connections with each other, from a few to thousands of connections with each neuron. So the number of connections between them is vast. And what we don't know is how memories are encoded in those connections. So
can you explain to us, non-scientists, what do you mean by encoding, number one, and number two, why are these connections made? How does it originate? Is it like somebody makes a phone call and somebody picks up on the other side?
Well, it's a little bit like that. I mean, it is an electrical and biochemical connection. But the coding part is, if we use the word brain, BRAIN, we know that those letters put together encode the word brain, and we recognize that as the organ in our head that allows us to think and act. What we don't know is how those neurons that are making all these connections to their neighbors, how those encode memories. We know the memories are in there. If the connections are broken, the memories are lost. But we don't know what it means. Do you need to have 100 connections from neuron A to neuron B and another 50 to neuron C to remember that cat is spelled C-A-T? We don't understand that. And so that is the huge problem right now that is bugging us with this. I think at the cellular and molecular level, we have a fairly good understanding of at that very small level. And then there's a lot of work with imaging, looking at areas of the brain that are turned on or turned off in terms of, do they use a lot more energy with a particular activity? But those lack right now the resolution to be able to assign them to specific neurons. So that encoding process is just beyond us right now. So that's where we are trying to model that.
Thank you, Gary. So, Matthew, when we're talking about modeling the brain, we're modeling the brain already, but we don't have a total understanding of how these connections are made and in terms of creating memories. So could you just run through what are we doing right now in terms of the knowledge we have, and what are the dangers? What are the positives and what are the negatives?
Well, I think one way that we understand now is, as Gary was alluding to, is how a memory is formed physically within a neuron. But what we want to try and do is try and find out how the memory is actually formed in our brain. And we know that when you experience something, your senses experience something, those actually end up as new growth neurons.
Mhm.
The neurons will grow and divide and then form a particular structure. And that sort of structure of the interconnection between different brain cells, as well as other cells, which are called glial cells, which actually form supporting structures. And they support and also chemically maintain the neurons. There's all this sort of massive different types of structural matter. And so when you come across the same experience again, this immediately lights up the same group of neurons. So
Is that how dating apps work? Is that how dating apps work?
I think Gary will talk that a little bit later on, I think. But this is how a memory is formed and how you recognize something when you come across it again. And this is what we're trying to find out. We're trying to understand how those actually happen. And the organoids, I think, will be able to do this because the nerve cells in the organoids are actually build themselves up into structures that have a sort of a second order structure, and they actually do communicate with each other in a particular pattern. And so we want to try and find out how that happens.
All right, Gary. As far as practical applications right now, what are the positives and the negatives?
Well, I think, when we're talking about modeling the brain, you're talking about trying to figure out how to predict what people will do. I mean, that's one practical application is that people look for that. And, so we can look at it, on the smallest structural levels of the cells and the molecules and so forth. And you can also start modeling it from larger imaging and looking at from both ends. But there are some people out there that, as Matthew or you alluded to just a moment ago, who are already trying to make predictions about the way we think. Dating apps clearly do that. And so they're taking information about us, about our behavior, and trying to look at all of those and trying to match us with another person. Or advertisers are looking at your behaviors and trying to predict your other future behaviors. And the scientists are also doing very similar things, I think, by looking at which parts of the brains turn on and off, and with various thoughts and actions and so forth, as Matt was just saying. And, there are ways of doing that, but always trying to get better and better resolution on it. For instance, there have been people who have looked at small sections of dog brains where they look at 1,000 cells, not in an organoid, but in a section of a brain, and try to just, rather than, they're just going to detect the signals they get from that particular subset of cells in response to showing the dog a bone or something like that, or food or something. What happens, as Matt said, and can you see that those same cells, those same kinds of electrical activities happening no matter how many times you do that? And then you can start learning how those neurons interact with each other. There are some practical applications of this coming down the road, maybe. People are looking at ways, for instance, with people who have lost the ability to speak, can you figure out what their thoughts are and translate that through some artificial interface into giving them the ability to speak or take an action? And these things are called imagined speech. It almost sounds like science fiction, and it's sort of in its early stages, but this is real research where they're trying to get enough resolution on those different areas of the brain that light up so that they can match those by following, for example, the difference in subtle changes in the musculature of your face when you're speaking. So these people might be able to move their face, move their jaw, and so forth, almost like reading lips, but also reading all the other muscles and the way the eyes move and so forth, and somehow collecting all that information and changing it into speech. If I could give one more example, there are efforts also being made to be able to communicate between the brain and a severed limb, for example, where there's a prosthesis in place, so that movements by the brain telling other muscles to move, those being picked up, and that information then being used to inform the motors in a prosthesis. So, but once again, it all depends on being able to collect enough information about the different areas of the brain and how they work. Because as you can imagine, this is, to figure out what a small child has 1,000 words.
Mhm.
We probably have 20,000 or 30,000 or something. To get that kind of resolution would be very, very difficult.
Right. Gary, I'm interested in this interface you're talking about. Is this an algorithm, or is it physical, or is it chemical, or a combination of all three, or anything else?
Different ones are different. The one I just was reading about recently was at Michigan, and they had taken, this is to power a prosthesis, and they had attached a small bit of muscle tissue to the end of the severed nerve. So then now you've got that connection between the nerve and the muscle back, at least a small piece of muscle tissue. The brain can then be taught to fire that muscle, and then the information from that firing of the muscle can be picked up by sensors in the prosthetic arm that tell the arm to move in different ways. So in that sense, it's sort of a tissue sort of thing. Other ones are looking at the almost like having a camera in front of your face to try to pick up the movements of your face and so forth. Very difficult because changes in lighting, as you can see on my face as I move, different camera angles, it's awkward. Others are trying to look at more of the electrical movements. They put an electroencephalograph where you put sensors all around the head and then try to pick up enough of those different areas of the brain, to teach them to be able to then use that could be picked up by other sensors and go into some sort of an artificial voice box.
All right. Well, thank you so much for coming on today. But before we leave, I want you, Matthew, first, and then Gary second. We talked about where we're at right now and what we're developing in the near future. Just let your imaginations run a little wild, and can you tell us where you think and what we're going to be able to do maybe in the next 25 years or so?
Well, as I said, I think these organoids are going to be such an incredibly useful tool down the line to understand how the cells, the different neurons and the glial cells communicate with one another in order to process thoughts. The one caveat I have is that I don't think you'll be able to ever build a physical, a biological model of an actual working brain, because everybody's brain is different from each other. You learn different things differently. You have different experiences. Your senses are, maybe some of your senses are more heightened and more aware than others. And so the memories and your experiences end up as a particular structure in the brain, which is unique to you. So I don't think we can copy that, but we can build this artificial intelligence, which is now being developed. They use a process called machine learning, in which the computer program is essentially given repeatedly different scenarios and essentially then tries to perceive a pattern. And that's, of course, what our own brain is doing. Our brains are very, very good at pattern recognition. And I think pattern recognition is all part of having a certain high level of intellectual abilities. Just think of an octopus, for example.
Right.
So it's pattern recognition for a machine to learn a pattern and then be able to then perhaps predict, given us a few smaller circumstances, and to determine how that pattern is going to end up. And this, I think, was part of your original idea, Bernie, about sort of where the future can be going.
Right. Gary?
Yeah, I would look to a more maybe practical end of this in a sense rather than the science fiction one. And that is that I think that an awful lot is being learned at certainly at the cell and molecular level. And I think that, I think that bodes well for diseases like Alzheimer's and stroke and spinal cord injuries. I think that those things are, because we know as much as we know right now, and the work in the labs is almost like science fiction, a lot of it. And I think those diseases, for example, I have great faith that it may be too late, Bernie, for you and me and Matt in terms of Alzheimer's disease. But I think that for our children, it's quite likely that our children may not have to worry about those so much because I think.
Well, I think on that positive note, we need to conclude. It's important for us to look forward to something good. So thank you so much for coming on today and talking about this. I hope to have you back on again soon. And we will certainly get together and talk about what we want to cover next time. Thank you very much and have a good weekend, everybody.
We're looking forward to it.
Thank you.
Thank you, everyone.